Thank you for inviting me. It's always fun to come to New York, and you can't hardly beat this venue, that's for sure. So I'm going to kind of give you a whirlwind, because in a half an hour it's difficult to talk about all the technologies and get into much depth. But I'm going to try to sort of whirlwind this and take you through, so that Santi Muni, who's coming up next, is going to present to you a bunch of the data using the technologies that I'm going to show you. Okay, so I guess you all know what's happening, of course, is that genetics is infiltrating all parts of reproductive medicine, from population screens, where we get tested by various companies to find out what genetic disorders we carry as heterozygotes or homozygotes, and gene panels where they actually look at specific disorders that followed the old Ashkenazi Jewish population type models, are sort of giving way to next-gen sequencing because of cost primarily. Get lots more data with next-gen sequencing. You're not looking at specific mutations. You are looking at entire exons of genes. We're going to talk about preimplantation, PGD for genes, PGS for chromosomes. And then, of course, lots of things are going on in the prenatal area, and all of this molecular work has moved into amnios and CVSs, and also non-invasive prenatal testing of free fetal DNA circulating in mom's bloodstream. And then, of course, we've got postnatal pediatrics, where testing is done now on newborns that are a clinical enigma. No one can figure out what the problem is with the child, and the cost of next-gen sequencing is now gotten to a point where it's about the same as a day in the neonatal nursery in a typical hospital in the United States. And so with the turnaround times being as rapid as they are, this is actually helping doctors in clinics with newborn kids that are kind of funny looking and you don't know what's going on. So the technology is less expensive, which is really the engine that's driving most of this. It's certainly more comprehensive. We can look at more and more things, which is a double-edged sword, because we all carry stuff. And when you find out what you carry, it creates anxiety and stress. And it's not just the cost of the technology, it's the cost of talking to the patient, or the cost of explaining it to them. We take an oath to do no harm, and here we are giving them lots and lots of information that does create anxiety. And so how do we take this profound amount of new knowledge and take it to the patient in a way in which we don't do harm, in an ethically responsible way? Certainly very rapid, and now it's becoming routine medicine. We always hear about genomic medicine. So we've come a long way from this first paper that Alan Handyside and our groups at Hammersmith Hospital in Houston at Baylor put out, where we actually were able to test for one mutation in one gene, that cystic fibrosis. And it seemed like science fiction at the time, but now here we are, where as of December, we were up to almost 40,000 PGD patients in our group, over 280,000 embryos for over 1,100 different gene mutations. So it's gone a very long way. In fact, there's really no molecular limits to what we can test for. We used to test for them using STRs, and you heard the guy from Harvard this morning talking about STRs, where you'd run these capillary electrophoreses, and you'd look at these polymorphic markers through a family. Each one of these cases was frankly a mini research project. You had to get the blood samples or cheek swab samples from the parents. You had to analyze those. Sometimes you had to go up to the grandparents. You'd find informative genomic markers. It was a lot of work. Well, the technology that would allow us to look at all of those genes did exist, but it was just not cost effective. And so new technology has really come along, and those of you that don't know about karyomapping, I want to explain it to you, because some of the concerns that was mentioned this morning about allele dropout and all that business, that's really kind of no longer an issue with karyomapping. I don't really like that term. Patients don't understand what it is. We call it ultra PGD, but whatever you want to call it, what it is it's the latest technology in single gene testing. And I'm going to try to explain it to you real simple, not because you guys don't know any genetics, but because if you try to explain this to a patient, you need a metaphor. And this metaphor seems to work pretty well. If you imagine, we'll just take one chromosome out of your set. So let's take chromosome 1, and we've got mom's chromosome here, and we've got all these genes, which are like cards in a deck. And we've got dad's chromosome 1 over here, and he's got all of his genes, and now we're going to have the two of them get together, of course, except that nature doesn't shuffle like a Las Vegas dealer, where we get a gene from mom and then a gene from dad and a gene from mom and a gene from dad. Oh, no. Nature shuffles more like a child would shuffle, where a whole bunch of genes from one parent and then a whole bunch of genes from another are crossing over. And, of course, these multiple genes, they all stay together. This all happens in prophase in the gamete in meiosis, and of course, it's called genetic recombination. And Thomas Morgan, the famous British geneticist, figured this all out, actually took pictures of it with his primitive microscope. You could see the crossing over. They drew out exactly how the chromosome from mom and dad would cross over. And why am I telling you all this? Because we use that as a tool to understand where the genes went in meiosis and mitosis. So just to remind you, we all have polymorphic markers in our genome where you and I are different from one another about every 1,000 bases in our DNA. So if we have every 1,000 bases, we've got a change, and we've got 3 billion bases in a haploid genome, or 6 billion in a diploid genome. Divided by every 1,000, we've got about 3 million single nucleotide polymorphisms that we can use as a molecular fingerprint to tell you from me. Okay? So we use that along with the recombination process in karyomapping. And this is all done on little teeny-weeny micro beads. So these little beads are really tiny, and they're sitting on an array, and they're held in there with old Van de Waal forces. They're not glued in there or anything. They just stay there, and you can wash the slide. The little bead is so small, it just stays in there with Van de Waal forces. And each one of these beads, think of it like it has grass growing out of the bead in all directions, except instead of it being grass, it's little artificial oligonucleotides. And each one of those little oligonucleotides represents a specific DNA sequence at a specific address in the genome. So this little bead might have some markers at a certain spot on chromosome 1, and this one might have a certain place on chromosome 9, or whatever. And if we've got millions and millions of these, in effect, we can interrogate on this array, we can interrogate nearly the entire genome. So to give you an idea of size, would be the size of a red blood cell, and this is how many of those you can get across the diameter of that cell. And they're just all on an array, kind of like the arrays we used to print, only these are now done on beads. Okay, so we've got a chromosome from mom up here at the top. There's the centromere, there's the telomere, and we've got a gene of interest in here. And for the sake of this discussion, we've got one gene that's perfectly normal, and we'll give this person a mutation in the other gene, so this individual is a heterozygote if this is a recessive disorder, is a heterozygote, or if it was a dominant, then this individual carries a mutation. And we follow, with those little beads, we follow the SNPs throughout the entire genome, but we're only particularly interested in this region right here by the gene. So we follow these SNPs from mom as it goes on this side of the gene all the way across over here, and then, whoops, we have a recombination. We have a genetic crossover, and we see the SNPs from mom's DNA now down on the other side. And at that spot, the crossover goes the other way. So let me sort of show you that in a different way. Here's a diploid cell from dad in the testis, and he got a chromosome from his dad, we'll give it blue, and a chromosome from his mom, we'll make it red, and there's the centromere in there. And now, when he makes his haploid sperm, these are going to do crossovers. And you can see that this section of that sperm's chromosome came from dad, then a section from mom, then a section from dad, etc., all the way down. And the other sperm that he's going to make from this diploid cell is going to have crossovers too, and this is random. Well, not completely random. There actually is some hot spots of recombination, but basically this is sort of like a Google map of each cell, or each sperm, or for that matter, each egg. Because mom's going to do the same thing, except instead of making 2 gametes, she's going to make 1 in a polar body, and we're going to be able to see those recombinations in hers as well. The take-home message from this is every single, there must be at least 1 chromosome crossover in every arm of every chromosome. And if the crossover doesn't occur, then you have early sister chromatid separation, which is the most common reason for aneuploidy. So there's arguments about why does this happen and what's driving all of the molecular machinery to make this occur. We heard some great talks this morning about this and about spindles. And we don't exactly understand it all, of course, but it could be energy from mitochondria, and I think Santi's going to talk a little bit about mitochondria. But anyway, you must have a crossover on every arm, and we can use this to follow the genes in your patients. These things are called haplo-blocks. So let's take an example. Here's a genetic ideogram of chromosome 7. There's the centromere, here's the long arm, there's the P or petite arm, and right in here is where the cystic fibrosis gene lives. And now we'll take dad and mom, your patients, and we'll give him, for the sake of this discussion, we'll give him a mutation in the cystic fibrosis gene, so he's a heterozygote. You found that out because he's got a family history of it, somewhere in the family of a child with CF or something, or because they were screened with one of these population screens. And we'll give her a mutation as well in that gene. They could be the same mutation, they could be different, it doesn't really matter. So they're both heterozygotes. So we're specifically interested in this little region of the chromosome. But we're actually obtaining information from every place on every chromosome at the same time, in the same test, at the same cost. But for your patient, we're only particularly interested in what we know that they're at risk for. So we hone on into this area, and we have an embryo that obtained a sperm that came from a sperm with this crossover map, and an egg with this crossover map in which the pronucleus had this one. And you can easily, by looking at all of those SNPs on the array, you can identify that this embryo inherited the haplo-block that comes along with the mutation from mom and inherited the normal copy from dad. Here's a different one in which it inherited the normal from both, so homozygous normal. Here's one that got the mutation from both. And they're having different mapping crossovers, but in the area of concern, you've got 50, 60 different SNP data points. It's not one. It's not like we had to go in there and sequence it, and if we had allele dropout, we'd make an error. Or the old days of STRs where you had to spend 2-3 months developing the STRs to do the case, because these SNPs are just sitting on the array. So this embryo is affected. This is a heterozygote as well. Now this one, about 3 years ago, we would have told you don't transfer that embryo. It's dangerous. We know that it has the mutation from mom for sure. That haploblock is consistent with the mutation. And there was a genetic recombination in the critical region of the CF gene. And that region, we don't know exactly where that is with STRs. So danger, danger, don't transfer that embryo. But with this, the fidelity of this technology, we can identify exactly where that recombination happened. And it happened, in this example, it happened in an intron away from the mutation far enough that we know that the haploblock of concern, the one where the mutation might or not be, isn't there. So that embryo is perfectly safe for transfer. So the technology has allowed us to get more information quickly, at less cost, and with more reliability. So to give you a sense of this, in 2015, our group, and there's a lot of groups now that do PGD, in our group we did 699 single gene cases. 84% of those cases were with karyomapping ultra PGD. 16% of them were using STRs. Now, why would we do any STRs? Well, most of it had to do with the fact that these were couples from overseas. They would fly to a clinic in the United States or Canada. They would want to have their biopsy on day 5, and they wanted to do a transfer on day 6 so that they could go home with an embryo on board. Even though they knew it would be better to freeze and do a transfer later, if they had multiple normal embryos, let's take a shot, let's take one back. And the karyotype mapping takes more than overnight. By the time you transfer the sample to the laboratory, you can't get the results by tomorrow morning when the window of implantation might still be open, and so you'll have to use the STRs. So it went from 84-16 in our group. Now, through September of this year, we've done 743 cases, 91%. So we've gone from 84 to 91% with ultra PGD. We've gone from 16 down to 9% STR markers. This is not because of science. This is because of convenience. So if you look at all of the pre-implantation testing that we do in our lab, 26% of the cases that we do were PGD only. 74% of them were PGD and PGS. The doctor biopsy, the embryologist biopsies the embryo, and the patient says, well, if we're going to do all of that, we might as well test both genes and chromosomes at the same time. So of those PGD cases, 74% of them asked us to do chromosomes at the same time. But about a quarter of them just said, no, test me for our thalassemia or whatever we're at risk for. We don't care about chromosomes. Oftentimes, we test this serially to try to keep the cost down. So we'll say to the patient, look, you're at risk for a dominant disorder. Statistically, half of your embryos are going to be affected. Let's test those first, identify those that don't have the mutation, and then do PGS on just those so we can keep your costs down, as an example. And so far in 2016, we've got 574 cases of PGD single gene plus PGS chromosomes. So it's routine if the patient and the clinic want it. So the take-home message so far is that in testing for Mendelian inherited disorders, there's no technical limitations anymore. Used to be there was, but not really anymore. There isn't any, except for disorders of imprinting. Not that we can't test for imprinting, and again, we heard a wonderful talk this morning about imprinting. Not that you couldn't test for it. The problem is, unless you really understand the clinical outcome of what you're testing for, how do you make a predictive response to what you find in the laboratory? So we've got lab data, and we know we have clinical information about abnormalities in imprinting, but connecting them together at the time of an embryo, not very many people, in fact, I don't know anybody that's doing that. And we also aren't testing mitochondrial heteroplasmy, not because you couldn't count how many mutant mitochondria are in the cell of the biopsy, you could, but because if the result comes back, it's 31% mutant, what do you tell the patient? Who's got the courage to tell the patient that's going to make a normal baby? And so until we have better clinical correlations, better clinical genotype-phenotype correlations, we're sort of stuck. Not because of the technologies, but because we don't understand enough genetics in medicine and early development. One of the things that we're doing that I thought I would throw in here because you heard a nice talk this morning mentioning fertility preservation. I want to tell you one story. So we've done a lot of these, but I'll give you an example of one. We had a woman aged 31, and she was diagnosed with breast cancer, and she had screening done, and she has a mutation in the BRCA1 gene. And she was in Houston at MD Anderson Cancer Center, and she went across the street to an IVF program to have fertility preservation before her treatment. So they did gonadotropal stimulation, and they collected and vitrified 11 of her eggs. She then went back over to Anderson, and she had her chemotherapy and her radiation treatment, which almost surely would have rendered her infertile. And then 21 months later, they thawed 4 of those 11 eggs, and she went through PGD for BRCA, PGS for chromosomes, and she had a healthy girl. Then some months later, she wanted to get pregnant again. They thawed 3. They didn't have any embryos that were euploid and BRCA-free, mutation-free, so there was no transfer, no pregnancy. Then they finally thawed the remaining 4, and she had a healthy son. So here she is, and here is her daughter, and here's her son. Both of them don't have a BRCA mutation. Both of them are chromosomally normal. And she has children after her chemotherapy. And we've now got, actually, it's more than this now. I've stopped updating the slide. But we've got over 250 now children born avoiding cancers, not just BRCA, but avoiding cancers this way. So we can test for cancers. And these are some of the ones that we test for on a regular basis, gene mutations that can cause cancer. In a half an hour, I won't take you through what they are, but there's plenty. Okay, so what's the new limit? Well, it's not molecular technology anymore. It's not. It was. Now the limit is the biology of IVF. It's the stuff that we've been hearing about all day. And what's going to happen when we've got lots of eggs that we can fertilize from in vitro maturation, etc. Okay, shift gears. I want to talk about chromosomes for a minute, and I want to talk about not so much the technology of that, but the issue that's going on with mosaicism that everybody's talking about. So first, let's just quickly go through the technologies just to make sure you've got them all in place. So when PGS started, of course, it was using FISH, and you had one fluorescent signal per chromosome. You had one data point, and you would read that data point under a microscope, a very subjective test. And you would instinctively err on the side of caution. You didn't want to make a mistake. And so there was a high false positive rate. Lots of normal embryos seem to have been thrown away by lots of groups who really weren't super experts at this. In the really good labs, it actually did work, and pregnancy rates would go up. But the miscarriage rates were high because of the false positives, or false negatives. You get pictures and you didn't know if there were 2 or 3 overlapping signals. It took a real expert to be able to read these things. So you had 11 signals in FISH because you couldn't even look at all the chromosomes. Then along came a qPCR technology, primarily done by a guy named Richard Scott. And what they did was said, well, 1 isn't very good. Let's do 4 signals per chromosome. So they identified 4 places on each chromosome, and there's nothing special about those places except that they're not polymorphisms. They don't change. 4 signals per chromosome. And why did they pick 4 and not 5 or 6 or 7 or 3? It's because 4 signals per chromosome times 24 chromosome is a convenience for the lab because it's a 96-well microtiter plate. Okay. So you're going to do a little PCR reaction at every one of those 96 spots in that little biopsy, whether it's a single cell from a single blastomere or a trophectoderm biopsy. You're going to do this PCR of every one of these things. And then you're going to take all of those, and you're going to you have them in a real-time quantitative PCR machine. And so as the signal, as the amount of amplification goes up, the ones that are coming off late are the ones that have less target material at the beginning. So these are the monosomes over here, and the trisomes are coming up early, so they're over here, and you need a computer to deconvolute all of those data. Then we have microarray CGH. You've all heard about this. You've all seen these pictures. The nice thing about this is that now we've gone from 96 to 2,700. We are looking as well at all the chromosomes, not just 11. And you actually, because of the way these were designed, these probes are evenly spaced all the way down the chromosomes. So you actually get about, depending on the size of the chromosome, but on average about 125 data signals per chromosome. So you can see it's getting lots better. And these are just red, and you would see those plots that you've seen over the years. Now there's a technology out there called FAST-A. This is the technology primarily done, in fact exclusively done, by Good Start. You've gone from 2,700 to 26,000, so that sounds like it's a whole lot better. And in effect what you're doing is you're analyzing your DNA for something that naturally is there, and that is this. Over eons, our DNA has picked up viral sequences. And these viral sequences, which are called transposons, and it was mentioned this morning as well, they're all over our genome. There's chunks of viral DNA. And once they got in there, they started replicating themselves. inside the genome. So there'll be whole stretches of this viral genome that's in there. It doesn't seem to do anything that we know about, but it's just there. The most famous one, the one that we have done the most research on in the biological sciences, is something called line 1 transposons. Now, as they duplicated themselves, they would then evolve independently, so they would create a mutation or two in the sequence. So now all of a sudden you had a sequence that had a unique change in the DNA, so it was like a map. So what Good Start does is they do an amplification of those 26,000 places across the chromosome, and that works actually quite well, except that what happens is because these transposons are not uniformly distributed down the chromosome like the microarrays were specifically designed to do, you've got lots more signals, but you have whole missing areas where you're not getting any signals at all. This is a very inexpensive test, so it's very easy to say, oh, I can do this for $75 or $100 a sample. But the problem is you're missing large amounts of information. If you're only interested in counting chromosomes, that might not be such a bad deal, except the false positive and false negative rates of this technology. But in any event, the British laugh and say, mind the gap. These are the areas where we really need to be paying attention to. And for reasons that I don't understand, I don't know if anybody who's a line 1 expert understands, but the transposons seem to be there a paucity of them on chromosome 21. And so in a chromosome that you really want to get a lot of information on, you're missing lots of information. SNPs, single nucleotide polymorphisms, there's hundreds of thousands of SNPs on an array. Some of them are informative, that is to say mom and dad's DNA are different, and so you can use those in order to understand what you're looking at, because if they're the same, you don't know anything. So you get down to about 30, 40,000 SNPs in the genome, and they're evenly distributed, so you get a nice signal of about 1,300 data points per chromosome. And you can easily identify, you get all this data, where there's homozygous for dad, homozygous for mom, and then a lot of heterozygotes in the middle. And you and I would look at this and go, how the hell can you read that data? But the computers can, and it'll easily see that there's a trisomy. Okay? So it works. One of the problems with it is you need parental DNA, so it's more like a PGD case. So you have to send in a blood sample on your couple ahead of time before you send the embryos. That's a downside of the technique. It's the downside of any SNP technique. So that's really difficult if you have any gamete donors in your group, because how are you going to get a blood sample from the gamete donor? Then we have scanning next-gen sequencing. This is a less expensive way to look with next-gen sequencing to look, and instead of looking at lots and lots, we're looking at about 100,000. So you see in a very short period of time how many more data points you're getting. And it's kind of noisy data, to be truthful, but you actually can pick up whole chromosome abnormalities as well. It misses subchromosomal aneuploidy, which is about 12% of IVF embryos, so that's a problem. And it's unable to detect chromosome translocations unless it's the whole arm of a chromosome, so that's kind of a problem. And it can't detect mosaicism at all in the biopsy, and you have to decide for yourself whether that's a problem or not. I'll give you my view in a minute. And you have difficulty distinguishing the data from background noise. So is this real? Is this a 3 megabase deletion or not? Or what's going on here? So there's a higher false positive with the technique. And then finally, there's the high-resolution next-gen sequencing. Now we're all the way up to 1.2 million data points in that little cell that you biopsy. Can you believe just a few years ago we were looking at 1, and here we are now at 1.2 million. You get about 130,000 across the genome per chromosome, and you get data that look like this, that you've seen these at meetings for a long time. It doesn't take anybody to be able to tell you that there's trisomy 17 in this embryo. This goes all the way up to the 3 copy, or sometimes they go all the way down to the 1 copy in a meiotic error. You get crisp, unambiguous data. It detects meiotic aneuploidy, and it can even pick up small copy number variations, like this is a 3 megabase deletion on chromosome 5 that causes profound mental retardation in Cree-Duchau syndrome, and you can pick that up with this technology. You wouldn't pick it up with any of the other ones. So as you get finer and finer and more information, you begin to make correlations of the data with the clinical phenotypes. All right. This is a picture of the southern sky from Kitt Peak National Observatory in Arizona, with the naked eye looking out, and it's a beautiful sky. That is the same sky. Nothing's changed except the detection technology. And that brings us to mosaicism, because now we have technology to see it. See, we've always known mosaicism's around. We see it in CVS for the last 35 years. We couldn't see it in a blastomere from a day 3 embryo because you only got one cell. And when you take a trophectoderm biopsy, it might be there, but if your detection technology can't see like the Hubble, then you wouldn't know. You've been transferring embryos with mosaicism since you did your first embryo transfer. You just couldn't see it. This is not a problem with the technology. This is a problem with our understanding of what the data is trying to tell us. So PGS was developed to test for meiotic errors in the gamete. Embryo mosaicism is caused by a post-fertilization mitotic event. Got that? So first, I'm going to just show you a quick set of slides to show you that this next-gen sequencing with high resolution can see it. So here's a normal female embryo, and you get data right at the number 2 because it's a diploid cell. Nothing's up here at 3. Nothing's down here at 1. Beautiful data at chromosome 1, 2, 3, 4, 5, all the way out. And then you've got 2 X's and no Y. Normal female. And here is a cell that has, it's male, so there's 1 X and 1 Y. There's nothing up here at 2. And you have euploidy all the way along until you get to here, where you've got monosomy 16 and trisomy 19. So let's just do some cell mixing experiments and see how easy it is or difficult it is for next-gen sequencing to pick it up. So if we take 10 normal cells, we're going to get this. 10 mutant cells, we're going to get this. Let's do 9 and 1 and 8 and 2 and 7 and 3 and see what we get. So, okay, so this is the mixing experiment. Here we are at 90% female and 10% male with the abnormalities. Now, you look at that, and I wouldn't call any of that abnormal, right? You just, even if you knew what you were chasing, probably you wouldn't pick it up. So the technology is not good enough to pick 1 in 10 cells up. Here's 20%. Now, what I want you to look at is I want you to follow the Y as it's moving up as we add more of these cells in here. And I want you to watch this 2 areas. So this is 20% mosaicism. This is 30% mosaicism. You begin to start seeing something. You see this has moved up. Here's 40% mosaicism. This has gone a little higher and a little lower as it's moving up to pure trisomy and pure monosomy. There's 50% mosaicism. See how this is going up? There's 60%, there's 70%, there's 80%, there's 90% mosaicism. This technology is so good that you can tell how many of the cells in the biopsy have this. So let's make a little cartoon about what might be going on. And let's just start on day 3 for the hell of it. So on day 3, we've got an 8 blastomere embryo, and it started with a normal egg and sperm. This isn't a meiotic error now. And a somatic cell in the group goes rogue. It has early sister chromatid separation, anaphase lag, something's wrong with the spindle apparatus, who knows? And now you've got 1 cell in there that's abnormal, and you've got the other 7 that are normal, and because of that, you now want to watch these chromosomes as they separate. So you're going to go to the daughter cells, and you've got these chromosomes, and we'll just do this as anaphase lag. The chromosomes go where they're supposed to go, and here we've got this one that's left over, and as cytokinesis comes, it ends up in this cell instead of this one. So we've got trisomy over here and monosomy over here. Nothing wrong with the gametes. This was a normal fertilization. All right. So when this now goes to the next cell division, we're going to have now 2 cells that are abnormal and 14 that are normal. And we're going to have 1 trisomy cell and 1 monosomy cell. And they're going to create patches on the surface of the cell or in the inner cell mass. The problem is you don't know where they go. But we're going to have a clonal patch of trophectoderm that has, let's just say, trisomy 21 and a clonal patch that has monosomy 21. Or maybe they ended up in the inner cell mass. That obviously we would all say that's probably worse. But we really don't know. We might all start out like this. We might all be low-level mosaics. So we've now got this blastocyst, and we've got these patches of monosomy cells and trisomy cells on the surface. And here comes in the embryologist, and he's going to do a biopsy. And they lay down the pipette somewhere on this sphere, and they're going to biopsy some cells, and they biopsy 5 cells, and all of them are normal, and everything's right down the 2-copy line. There's nothing up here at 3, and there's nothing down here at 1. You'd say that's a normal embryo, but these are here. You just didn't find it in the biopsy. So remember, a biopsy is simply a sampling. This is no different from biopsying a mass in the abdomen. And you pull out the sample and send it to the pathologist, and they say, oh, I don't see any neoplasia here. There wasn't any neoplasia in the biopsy, in the sample that was tested. It doesn't mean it isn't somewhere else. All right? Suppose they biopsy over here and get 5 cells. You're going to get the same result. And suppose they biopsy here, and one of the cells is abnormal because they just happened to set the pipette down next to the patch. Well, you can see that there's a 20% mosaic. The computer will call it for you. It's a 20% mosaic, monosomy 10, in that sample. And if you got 2 cells instead of 1, you can tell the difference, and there's 40% mosaic. What if you biopsied over here by accident? Now you see it trisomy, not monosomy. Wow. The same embryo, you get 2 different results depending on where you place the pipette. And you can pick up 40% trisomy as well. So we've modeled this, and my talk time isn't going to let me take you through all of this, but you can start to model this whether or not this anaphase lag or early sister chromatid separation happens in the division from 1 cell to 2, or from 2 to 4, or from 4 to 8, etc. And you get different amounts of mosaicism, of course, depending on when it happens. So we've hunted high and dry for this, and there's not very many of these. Bill Screwcraft has one. I think Santi has one at Reproductive Genetics, or maybe 2, and we've got a couple as well. And I want to just show you. This is rare. Don't think this is normal, but this is a hint that what I've been telling you might actually be true. So we did a case for a couple, and this is the profile that we got. All right, so there's an X and there's 2 X's and no Y. So this is a normal female euploid embryo, and they transferred the embryo, and the woman gets pregnant. This is a single embryo, single SET, FET. Then she gets a pregnancy loss at 7 weeks. And we were able to do next-gen sequencing on the abortus material. So we wanted to run this and see what we would find, and look what we found. We found trisomy 4 in the abortus material. And we went back to the original data and ran it several times and looked at it, and you might be able to convince yourself, maybe, you wouldn't have picked it up raw, but going back and looking, you might be able to convince yourself that there's a 10 or maybe 15%, we wouldn't have called it, monosomy here, mosaic, in this sample. So you're getting the reciprocal, just like I showed you in the little cartoon. All right, let me just give you a couple pieces of data, and then we'll move it over to Santi, who's got a lot more to share. So the overall rate of diagnostic outcomes in 15,000 biopsies that we looked at, this is what we see. About half, about 47% or 50% of the embryos are euploid. About 34% of them are aneuploid. And then we've got these others over here. What are those others? Well, about 5% of the embryos in this 15,000, about 5% of them actually do show some mosaicism, but they've got a full-fledged meiotic gamete heir in some other chromosome. So they do have mosaicism of a chromosome, but they're aneuploid, just like the rest of these. And then we've got a group that we call medium-high-level mosaicism, where about 40% or more of the cells are abnormal, or less than 40% of them. So that makes up these two groups. So if you add up all of these, you have about 18% of embryos actually show mosaicism, about 18%. But these, if we lump these over into here, because these also have, say, trisomy 1, and the mosaicism is on a different chromosome, now we're down to about 13% of the embryos that actually show mosaicism. Now, should you transfer those embryos? Well, what we call these embryos normal and we call these embryos abnormal, and this is the two sections where we can have lots of debating. And if you're very risk adverse, you don't want to transfer anything that looks like it might have mosaicism. And if you have a different philosophy in your practice, you might say, no, I want to give the patient the benefit of the doubt. So it's case specific. If this is the only embryo the patient has and it shows mosaicism, we have lots of data now that show it makes babies. You wouldn't transfer it as your first choice because we know that there's a miscarriage rate that's higher. One of the interesting things is that mosaicism, as you would expect, is not dependent on maternal age. It's got nothing to do with the gametes. But we see differences across different clinics. Why would that be? Well, some of this mosaicism might be iatrogenic. Some of it might be the way the biopsy's done, the way the laser's used, the culture media, who knows? But we definitely see differences from 13% to 27%. On my last slide, I just want to take you through some conclusions. The first is that the main goal of PGS is to find chromosomally normal embryos to transfer. That was the whole point of this. Meiotic heirs. We just now are getting mitotic heirs as well. We want a high success with a single embryo transfer. Who would have believed that we would be talking about 50, 60, 70% pregnancy rates with single embryo transfers just a few years ago? And we want to shorten the time to pregnancy for the patient. A biopsy is simply a sampling, just like anything in medicine. It's always been there. You've been transferring them all along, and we finally can detect it with high-resolution next-gen sequencing. The second goal is to reduce miscarriage rates. Richard Scott says something that a lot of us now copy, the quote, because it's the first time I ever heard it was from him. I don't know if he got it from someplace, but the quote was, there's actually something a lot worse than your patient not getting pregnant in the IVF center, and that is getting pregnant and having a pregnancy loss. That's an emotional roller coaster you don't want your patient to ride. And so if we can reduce miscarriage rates by detecting mosaicism, that can assist us in that small percentage of patients that I showed you on the pie chart. But genetics is only one tool in your toolbox. You've got a lot of other ones. So just because the genetics says it's normal doesn't mean we discard morphology and all of the other important things that this field has developed over time. And again, many healthy babies have been born from mosaic embryos. Don't be surprised. And if you rebiopsy the embryo and get a different result, don't be surprised. You now understand how it's happening. The pregnancy rate is increased over no testing, that's for sure. And the spontaneous miscarriage rate is higher if you transfer a mosaic. So if a patient has only one mosaic embryo and the rest are fully aneuploid, have a good conversation with them. Talk about it. Tell her it's not perfect. We don't think it's reasonable that it will make a normal baby. We can't prove it. We want you to have a CVS or an amniocentesis to be sure. But lots of babies have been born with embryos like this. It's not our first choice, but it's the only embryo you have. Let's go with it. Thank you very much.